Hey, what's your operating system? You probably already know that the information that eventually becomes you is stored in your DNA, but how does that information actually become action? I mean, for real, do you have any idea just how much effort it takes for your cells to keep you alive second by second, let alone day by day? This is a question scientists are only just beginning to scratch the surface of, despite it being over 70 years since we determined the structure of DNA and kicked the study of molecular biology into overdrive. Back in my day, with things like the Human Genome Project, we thought we could basically solve every problem once we fully decoded the 3 billion letters of DNA that form your blueprint. Folks thought DNA was destiny. But 30 years on from the first fully mapped human genome, it's honestly becoming more and more clear that your DNA is only really a contributor to this story. To oversimplify it a little, DNA is more of a storage medium than anything. Instead, this molecule might be far more important. Meet RNA, ribonucleic acid. In school, we treated this flimsy little strand like DNA's kid brother. But in reality, RNA might be the whole ballgame here. All of your complexity, all of the coordinated work it takes to keep you alive, at some point or another, relies on the transcription of DNA into RNA. And this is a massive process with more twists and turns than you would believe. Take the time to walk through how your archive becomes your action with me, and you will come away with an aching appreciation for the amount of effort it takes just to watch this video right now. It's time we start the long process of unraveling your central dogma. Welcome to your primer on how you really operate. And hey, welcome back to Clockwork, a show about biochemistry. And just so you know, this video is sponsored by Brilliant. More from them later, let's get into the actual video itself. This is episode 1 in our long, wandering series about DNA and how your genetic information becomes true cellular action. Make sure you check out episode 0 in this playlist to get a primer on your DNA and how it replicates before all of this transcription business goes down. Subscribe here to catch the rest of the series as it comes out. But for now, y'all, we have a lot to cover. Let's not waste any time. This series is about your central dogma, which is basically the core operating system your cells follow to go about the business of keeping you alive. This playlist will have 2 key chapters to start. First up, this video is going to cover Chapter 1, the transcription of DNA into mRNA. Chapter 2 is going to cover translation, or how your cells turn RNA into protein. And this episode is going to come in basically 4 main parts. First up, we're going to have to do a quick overview of the central dogma itself, just to understand the lay of the land for the rest of this series. Next up, we're going to have to talk about our 2 main molecular characters, that's DNA and RNA, and then we'll finally get into the actual act of transcription itself for the bulk of this video. And in part 4, we'll see how that RNA is edited after transcription happens, before it's sent out to do real work in your cellular machinery. Sound good? Let's go. Starting, of course, with what exactly is the central dogma? And I mean, it's hard to distill this down into a sentence. One way to think about it is that the central dogma is the story at the center of all biology. It's a topic around which everything else rotates. At least it was. It's maybe diminished a little bit in importance in the last 30 years or so. And this whole series is aimed at helping you unpack those complications and get a true, rich view of what the central dogma actually is. But in short, the central dogma is how information flows through your cells. In every single one of your cells, you have DNA, a complete archive of all of the genetic information you have from both of your parents. That DNA encodes information in the form of genes. When you need to do anything, your body reads your DNA to transcribe it into RNA. That RNA is then translated into protein. And those proteins, are the actual workers that go on to do the actual business of being you. So, at its simplest form, the information flow in your body is DNA becomes RNA becomes protein, right? Cool. That's the whole business of being alive, at least at a really high level. This series is going to show the central dogma in all its complexity in eukaryotic cells, going well beyond this simple chart. Eventually, we'll loop this chart back around and discover how proteins in our cells talk back to our DNA. And along the way, we're going to shake up this rigid view of how our cells work. Based on how you learn about the central dogma in school, you might think of a cell as a very mechanical robot. What you're going to see starting in this video is how remarkably fluid this process is, and that's the most important thing to keep in mind. Living systems are incredibly malleable. And to be clear, we're going to explore the central dogma in your cells specifically. As a card-carrying member of Team Eukaryote, you've got way more complex machine code than the bacterial systems most folks tend to learn about in regular school. Buckle up, because we're not pulling any punches when it comes to trying to understand the complexity at play here. And if you power through some of the incredible complexity we're about to deal with, you'll see how all your complications here aggregate to be kind of a superpower. And let me get you in the mindset for that first. Quick question. If every single one of your cells has a full copy of all of your DNA, then how does a cell in your kidney know to only transcribe and use kidney-related genes? What's stopping individual cells from making the, wrong proteins? Don't answer that now, just think about how that could be managed. But back to the matter at hand. I want to zoom in on this original central dogma chart here to see the full scope of our video. Today we're going on what seems to be a very simple journey. We're going from DNA to RNA. Specifically, we're going to be ending at a specialized kind of RNA called mRNA, or messenger RNA. However long that takes, mRNA is our end destination. If you watched episode 0 of this series, you know just about everything you need to about DNA for this video. Go watch that if you haven't. We'll wait. And hey, welcome back. Let's get to know our other main character before we kick off the real action here. Meet RNA, ribonucleic acid. Like DNA, RNA is a string of little nucleotide letters. The big difference you can see right up front is that RNA only has one strand. The double-stranded structure of DNA rocks for its function. Those two strands make DNA incredibly stable and very easy to copy, which is effectively DNA's main job. Don't break, get copied, be ready to be transcribed. Done. Nice. Meanwhile, the single strand of RNA makes it way more dynamic and also way more fragile. RNA is a molecule of action. The flexibility of RNA allows it to do some genuinely wild things, some of which we'll actually see in this video, others will have to wait for future iterations of this playlist. And you might also notice that the colors are kinda off here too. That's because RNA doesn't use the letter thymine. RNA has its own imposter thymine called uracil. It lines up with adenine just fine during transcription, and that's all that really matters, at least for this video. And for the sake of contrast, from here on out, I'll be using orange as our primary color for RNA. I think it's important we make our main character stand out for what's to come. Like I said, RNA comes in all sorts of shapes and sizes, but today we're focusing on the vanilla version of your RNA, messenger RNA. mRNA carries the information stored in your genes out into the cell to code for proteins, but getting there is going to be a whole thing. So, to make our view of the central dogma more accurate, this chart should actually read DNA becomes mRNA becomes protein. But how exactly does DNA get turned into mRNA? Which brings us to part 2, actual transcription, turning DNA into mRNA. Again, it's not the DNA itself getting converted, it's the information the DNA encodes for. This whole thing goes down in the same place where your DNA lives and gets copied. We're here in the nucleus of, literally any one of your trillions of cells at any given microsecond. The nucleus here is a big old ball of DNA bundled into these thick chromatin fibers. If we're going to turn DNA into RNA, we're going to have to find a way to stick this complex, RNA polymerase II, onto our DNA. Pol2 here is one of your primary molecular workers, and it is constantly, tirelessly assembling new strips of RNA as your cells need. During this process, RNA polymerase II is going to get loaded onto this DNA strand, and then a bunch of extra workers are going to open this DNA up and allow RNA polymerase II to read that DNA. The reading process is simple enough. This strand up top is what we're actually copying. Keep in mind the DNA's superpower is that it can be copied. Since these DNA bases have a complementary base pair that they can bind with, and since uracil can bind to thymine just fine, RNA polymerase II can use this DNA as a template to synthesize a complementary mRNA molecule. Once transcription gets kicked off, basically new RNA letters are going to come into RNA polymerase II's active site, line up with their complementary base pair, and bind to the growing backbone of our mRNA transcript. So we're not literally converting DNA into RNA. This DNA is being used as a template to assemble an RNA molecule. Awesome. Here's the thing though, RNA polymerase II is just a printer. It doesn't have any knowledge of what action your body needs to take at any given moment. It just needs to get attached to this DNA strand, activated, and freed to print. Meanwhile, the nucleus of your cells isn't just empty space full of DNA and whatever else. It's chock full of raw DNA and RNA letters floating around in space, just waiting to get added to a new strand. And again, going back to that DNA video, you'll also know that the act of adding a new letter to this transcription actually releases a ton of energy, which means that when you add a new letter to this RNA code, it releases a ton of energy, pushing the transcription machinery along with ease. If you don't manage RNA polymerase II, it'll just sprint down this DNA strand, Saquon Barkley style, unstoppable because of how much energy is released in this process. And if that happens, that's not a useful transcript. So in reality, instead of just being this simple printing process, transcription for you is an RNA polymerase management problem way more than anything else. Instead of starting with RNA polymerase II, we're going to meet a whole cast of characters that make sure transcription is on the right track. We're not going to meet all of the protein workers that manage transcription today, just enough of them to get us all the way from a random exposed DNA strand to a completed mRNA transcript. As we get further and further into this playlist, we'll fill in more and more details and see just how many managers there are, but I want to make sure we get a complete story here so we don't get overwhelmed from the very beginning. And if you want to get a better understanding of just how complex this management problem is, I really recommend you check out this video's Clockwork Book Club entry, How Life Works by Philip Ball. Ball has pulled off an incredible achievement with this book, distilling decades of research into a solid page-turner that explains our new understanding of all of the details surrounding the central dogma of biology. A lot of how I'm framing this series will heavily reference this book, so you may as well check it out now if you want to get an idea of where I'm going with this series. How Life Works gets an easy 5 clocks out of 5. I wish Philip Ball was my boss and/or academic advisor, but I'll just be satisfied with hawking his books for now. Back to the video at hand though, let's actually transcribe this DNA segment. Here are the steps we're going to go through. Transcription comes in 5 not at all easy parts. We got pre-initiation, initiation, pausing, elongation, and eventually termination. Each one of these steps has up to dozens of molecular assistants, so get ready for a whole bunch of names, folks. This is going to be a party. Let's go ahead and light this candle. For this video, we've actually kind of skipped the first couple of steps here. Before DNA can ever actually get transcribed, individual DNA strands have to be loosened from their tightly packed chromatin form. We're going to cover how this goes down towards the end of this series because it makes a lot more sense once we've covered the actual basics of RNA transcription and protein translation. For now, let's put in the work to turn this DNA into messenger RNA. And let's get a quick lay of the land as we kick things off. Just to orient ourselves, we're going to travel this way when we start transcribing our DNA strand. Just like replicating DNA, we have to crack open this helix and move across these letters in order to build our RNA chain. Which brings us back to the setup. This gene is exposed and ready for something to bind to it. We're going to kick it off here. It's transcription phase 1, initiation. This gene isn't super special, and has some of the basic features most genes do. The actual coding DNA of this gene is more down here, moving off to the right. Upstream a bit is this region, where a bunch of thymine and adenine letters repeat a bunch. This is your TATA box, folks. It's one of the best named structures in all of biology. Oh, and wait, yeah, that's backwards. So if I flip this around, yes, now you see, TATA box, right? Okay, let's rotate back so we keep the same orientation the rest of this video is going to be in. Savor a beautiful label like this while you can, because it's about to get immediately buried. Repeated DNA letters like this are just catnip for specialized proteins that can bind to DNA. Here comes one right now, the instigator for everything that's about to come. This little butterfly-looking fella is, get this, the TATA binding protein. He's actually a smaller subunit of this beefy complex, General Transcription Factor 2D, or just TF2D. Get ready for a lot of acronyms that sound just like this. So 2D is really here to kick things off. 2D's entire job is to first stabilize this DNA strand and get it so that the TATA binding protein actually has a chance to bind to the TATA box. First, the front end of 2D is going to recognize a point just a little downstream of the TATA box, associate there, and then rearrange itself wildly to attach this yellow TATA binding protein. And flipping the view real fast, TPB will lock onto your TATA box and be used as the initial anchor for the scaffolding your cell needs to attach RNA polymerase II to this DNA. As 2D locks in place using the TATA box, it's formed a foundation we can build the rest of the pre-initiation complex on. Another anchoring factor, 2A, is also joining the party right here. 2D has now set enough of a stage for our main character to take their place. RNA polymerase II is going to slot in right here, while a new complex, TF2E, binds, which is going to make way for the rest of the party to start. For the most part, this is your pre-initiation complex, the first major complex built by your transcription team. Which means we're ready to go, right? Wrong. With the pre-initiation scaffolding fully assembled, our workers will rearrange a little while the final 2 elements slot in. At the same time this complex assembles, this massive unit up here is getting recruited as well. This is your mediator. Our mediator complex is here to make sure that it's actually 100% time to turn this DNA into action. This TATA box might have been exposed randomly thanks to, any number of factors. So our cells have another layer of regulatory control built in here. Basically, proteins will bind to DNA segments called enhancers way upstream of the actual gene that needs to be transcribed if it's actually go time. Through a mechanism that scientists are still trying to nail down perfectly, those enhancer elements bind to this mediator. If our mediator gets the correct signals from these enhancers, it will help organize the final steps of the pre-initiation complex assembly. Basically, the mediator is going to help another transcription factor, 2H, into the exact correct position so it can help activate this whole process. It's going to do this by binding to one of the most important and weirdest parts of RNA polymerase II. This long, wiggly string at the end of pol II is the enzyme C-terminal domain. This is, in a way, pol II's like control rod. And hold up, just a couple of caveats here. This is just a small segment of this C-terminal domain. RNA polymerase II's C-terminal domain is an incredible central platform for transcription. What you're looking at is a very small section of this long, thread-like appendage on RNA polymerase. This, control noodle is made up of 52 repeating amino acid sequences. I'm only able to render about 4 of them, so basically 90% of this structure is just not visible in this video. The CTD's job is to get phosphorylated, basically to have phosphate groups added all across the serine amino acids along this structure. During this whole process, as different regulators are needed, this C-terminal domain have different segments get phosphorylated. Meanwhile, the CTD is so long that it can actually act as a holding scaffold for regulators to attach to as they wait their turn to affect the transcription process. Just know that each step of transcription changes the phosphorylation setup in a subtle way. And that's what makes the mediator important. It looks like the mediator's job is it will hold on to the C-terminal domain in a way that TF2H can kick off the first wave of phosphorylation and activate transcription. Let's zoom in to the actual DNA side of this process. 2H here is going to help unwind our DNA strand and properly spool it into the active site of RNA polymerase II. Basically just by shoving it backwards. We're now in the open state of this pre-initiation complex. Once it does, all bets are off. This complex is now fully activated, and the energy from adding new letters to that RNA chain will start moving the core transcription machinery rapidly. Which means now that we've met all these characters, we're going to instantly leave them all behind. Nice. And yeah, we're on our way. With RNA polymerase II released, initiation is over, and we're ready to jump into the next phase of transcription. But before we do that, let's check in with the sponsor of today's video. Today's video is brought to you by Brilliant, where you learn by doing. For me, I love learning by consistently incrementing my way through a topic. Even something as immense as transcription is learnable as long as you tackle it in bite-sized chunks. That's what makes Brilliant great. It's a learning service with in-depth and incredible lessons that can gradually level up your understanding of the harder sciences. I've been having a great time going through Brilliant lessons on computer science, shoring up my foundations. Each Brilliant lesson is filled with hands-on problem solving that allows you to actually play with concepts. This is a method proven to be 6 times more effective than watching lecture videos. So, while you're establishing a real basis of real knowledge on specific topics, it also helps you work those cognitive processes to help you become a better thinker overall. More importantly, Brilliant is an incremental platform which will help you develop a powerful daily learning habit. You're not going to learn data science or calculus in a single day, but you can incrementally chip away at the foundations of these problems until you can wrestle with the more advanced concepts. If you want to try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/clockwork or scan the QR code on screen. Or you can click the link in the description. You'll also get 20% off of an annual premium subscription. Give them a shot. You genuinely won't be disappointed. But for now, let's go ahead and get back into the video at hand. RNA polymerase II has been released, so what happens immediately after that? Turns out, it almost immediately gets stopped the second after it started. Awesome. And hey, we've come to the surprise third stage of transcription, pausing. Brought to you by this charming fella, NELF, the negative elongation factor. As this little baby RNA strand finally starts poking its little head out of the core RNA polymerase, for now, it's actually very important to pause here because we need to process this RNA before it gets too long. A single strand makes RNA versatile, but also fragile. Our mRNA here is also getting ready to be shipped into the cytoplasm of our cell proper, where it can get blasted apart by all sorts of intracellular goings-on. So we have to immediately start packing this RNA to ensure some stability here. NELF is here to tackle pol II long enough so that these yellow RNA capping enzymes can add this little methylated cap on the front of our growing mRNA. This cap is going to keep our little messenger stable, and more importantly, this is also something that our cells use as a kind of ID tag. mRNA is the machine code of the cell, no matter what. If an mRNA strand is loose in the guts of one of your cells, it's going to run into an army of ribosomes and then get translated. Therefore, your cell has a ton of internal defense systems to attack and destroy foreign-looking mRNA. And all this little cap does is block the RNA chewing machinery from accessing the rest of this chain. So honestly, it's a good thing that this is the very first modification we get hit with. So this immediate pause and arrest is just built in to guarantee that we don't run away polymerizing an mRNA molecule that will just get chomped up by your cell's defenses once it gets shipped out. Awesome. With the cap on, our capping enzymes are going to move on to the next baby RNA they need to process. Now, sometimes transcription will just stop here if your cell isn't super ready to make whatever protein this region codes for. NELF down here is actively holding on to the complex, so there's this tension building inside the stalled polymerase that could kick the whole complex off our DNA and kill transcription before it even begins. Unless this really is DNA our cell has tagged for transcription. Let's pan downstream a little bit, where we can see one of the histones that pack DNA into tight little bundles. When we definitely 100% want to transcribe something into protein, these histones will be tagged and altered, allowing this tiny little worker, PTEF-B, to bind. These histones usually are pretty close to where our initial stall happens, so sure enough, PTEF-B is going to be able to hop off this histone and bind to our stalled transcription complex with NELF. PTEF-B will use this scaffold to drop its own phosphorylation onto polymerase II's controlled stick, and that's going to change all these shapes just enough for POL2 to kick NELF to the curb and finally, truly, get this transcription party going in full swing. Welcome to the next phase of transcription, elongation. PTEF-B has changed things here enough that a massive new cast of helpers can join POL2 and stabilize things enough to get us through most of transcription. Every one of these blue workers is here to either stabilize our growing mRNA or to help unwind the histones that store our DNA. Let's go ahead and see how that works. And you know what, it only took us most of this video, but this is what the majority of transcription looks like. But yeah, let's just take a breather and allow elongation to play out for a sec. We really had to fight our way through a whole army of regulators just to get here, didn't we? Again, talking about the regulation of RNA polymerase II is so much more important than just the basics of transcription itself. So many of the fuzzy details that make us so rich and adaptable live in these dozens and dozens of regulators that keep our cellular machinery on the right track. And I really want to make it clear that I'm barely scratching the surface here in terms of all the assistant proteins that could be deployed to every possible interaction of transcription initiation and transcription elongation. I'm showing you an incredibly narrow, yet complete from start to finish instance of transcription. The goal of this playlist is to help you stand on a complete foundation and then fill in more and more detail, revisiting every facet of the central dogma. The complexity really is the point here, and now we have an excellent jumping off point to explore so many vital cellular processes moving forward. But look at that! We've passed into the 3' untranslated region. Our gene technically is done being transcribed. We're not making any usable code anymore. Let's lock in for the final phase of transcription, termination. RNA polymerase II has crossed out from the coding region of this DNA and has hit the end signal. We are now in the 3' untranslated region. As we pass into the untranslated region, all the lovely elongation factors here are just going to break off from POL2. Maybe not this dramatically, but, you know, work with me here. At the end of every protein coding gene, there's this little DNA sequence that's here to stop everything. Behold, the polyadenylation signal. The polyadenylation signal is absolute catnip for regulatory proteins. Specifically, a group of workers led by CPSF will bind here, cut our raw RNA at that polyadenylation signal, and help ship this transcript off for final processing. Once our fresh transcript has been snipped off, we're kind of in trouble. Again, RNA polymerase II does not know that transcription is over. It's just a printer. Polymerase is just gonna polymerize, so it'll keep going as long as it's allowed to. But, of course, duh, we have a way to manage this problem. Remember, that methylation cap we added to our RNA at the beginning is mainly there to block RNA-chewing enzymes from destroying it. But now, RNA polymerase II's C-terminal domain is just way too phosphorylated for those negative elongation factors to come in and stop things again. So now snipped and missing a 5' cap, this junk RNA is just begging to be chopped up. And incredibly, there's an RNA-chomping enzyme ready to party in this exact circumstance. In human cells, this worker is called XRN2. This Lockheed Martin-ass name is fine, I guess, but the yeast scientists did way better here, naming the equivalent enzyme RAT1. Which is sick because we're about to go rat mode to put a stop to transcription once and for all. RAT1 will grab onto this junk RNA coming out of RNA polymerase II and shoot along it like a missile. Once it reaches the end of that little RNA tail, it is actually the perfect shape to slam into the bottom of RNA polymerase II and physically knock it off of our DNA strand, terminating the transcription process. And this is called the torpedo model of transcription termination. It's over. We did it. Pol 2 is now free to go off and find new genes to transcribe, and our RNA transcript is free to get capped. If we go back to our freshly synthesized RNA, a new capping protein will join the party and add a whole bunch of adenine letters to the end here. Both ends of our RNA need to be capped to maintain stability. The main reason we have this poly-A tail at the end here, scientists figure, is to be kind of a freshness signal for this RNA. Even capped like this, RNA is still pretty unstable, but this long tail will degrade over time, getting shorter and shorter, so your cellular workers can assess the, freshness of any RNA that it comes into contact with and choose to destroy crusty old RNA that's past its use-by date. Maybe. But wow, let's just very quickly review the whole process and really lock it in. When it's time to make a new protein, your cellular regulators will loosen up the chromatin fibers containing our target gene. DNA has a bunch of signals that our transcription apparatus can bind to. We've got the promoter here at the start of the gene, and usually we have enhancers upstream that regulators will bind to. If the conditions are right, the TAT binding protein will bind to the TAT box, assembling all the TF2As, Ds, Bs, and Es needed to build a pre-initiation complex. The mediator will join the party and help stabilize the whole structure and help TF2H activate polymerase II. Pol 2 will then shoot out of the pre-initiation complex, instantly break down long enough for the new RNA to get a 5' methylation cap, and then get started back up with new modifications that will allow it to chew through all the histones that would otherwise block transcription. At the end, polymerase II is going to pass the 5' UTR and transcribe the polyadenylation signal, which will attract more workers that snip that fresh RNA off and drag it away to be processed. Now snipped and missing a 5' cap, this junk RNA is just begging to be chopped up by XRN2/RAT1. They join the complex and go rat mode, torpedoing into everything, pushing polymerase II off and fully terminating transcription. And we've done it, right? That's part 1 of the central dogma right there. Finished. We've gone from DNA to mRNA, right? But let's check out our freshly minted mRNA transcript as we gear up for final processing. Huh, that's weird. What are all these yellow bits in here? Welcome to the plot twist, folks. We've gone through so much already to get to this part of the journey. But remember, our goal for this video was simple. We're exploring the details of how your cells turn your DNA into mRNA. And that's what we've been synthesizing this whole time, right? Wrong. This strand right here, this is a eukaryotic special. As a lot of you probably know from school, bacteria actually follow this simple central dogma chart. DNA gets transcribed into mRNA, which then gets translated into proteins. Pure, simple, efficient, and it all happens in the same place at practically the same time. But that is not the game you play. You and every other eukaryote, from trees to baker's yeast, have a lot more work to do. Instead, what we've watched this whole time is RNA polymerase II synthesizing a special new class of chain, precursor mRNA. Of course, right? Pre-mRNA is just the raw information encoded in our DNA. For reasons we're about to explore, your genes are packed with air quotes useless little segments that don't code for protein. These are called introns, and we're going to color them yellow from now on. The orange segments actually do code for protein though. Those are called exons. All we have left to do is cut out these introns and stick our exons together so we have the right set of instructions for our ribosomes. So yeah, let's go back to this chart then. Plot twist, the real journey of this video is to use our DNA to synthesize precursor mRNA that will eventually get edited down into sleek, refined messenger RNA. To accomplish those edits, we will meet one of the most incredibly complex systems hidden in your cellular machinery. Your mRNA editor is this wild, roiling collective of workers called the spliceosome. How the spliceosome does its job is honestly hard to describe, but we'll cover the basics reasonably well in this video. But that's not good enough, because I figure you're like me when I first learned about this ludicrous complex in school. I figure you're practically screaming at the screen right now, asking, why do eukaryotes have to make things so complicated? And trust me, the why of the spliceosome will make this entire journey so incredibly worth it, because there's always a good reason for complexity. But let's not waste any time. Welcome to Plot Twist Part 4, discovering the secret superpower you have hiding in your spliceosome. I want to be clear here, because this video was already so long, we're only going to cover the very basics of precursor mRNA processing. We'll save a much deeper look into splicing and its machinery for a later video. You're gonna see why in a second. Either way, the spliceosome has two main jobs. It needs to recognize and bind to both ends of this intron, and then it needs to figure out a way to cut the intron out at both ends while stitching our exon segments together. Now, we've seen moments where proteins can bind to specific DNA segments or mRNA bits, like the polyadenylation signal. But what's going to reliably attach to both ends of this intron? In answering this question, we get to meet one of the coolest classes of molecule. Check out our cast of characters for this one. To splice mRNA, we're going to need subunits U1 through U6. Let's go ahead and zoom in on U1 and see exactly what we're dealing with here. This first piece of our spliceosome reveals an incredible truth of how your cellular activity is managed. Does U1 look kinda like weird? It might, because U1 is actually made out of both RNA and proteins. Specifically, this twisty strand is small nuclear RNA. Have you ever read that like 90% of your genome doesn't code for anything? Turns out a lot of the nothing coded in your DNA actually codes for little RNA machines called small nuclear RNAs. Remember from the beginning of the video, 1,000 years ago. Since RNA is single-stranded, it can twist and fold into lots of different shapes, just like the protein workers that supposedly power everything we do. Keep that fun little fact in mind as we move forward. So when you combine these wee little RNA twists with a protein assistant, like what we're looking at with U1 here, you get one of the most powerful and best named biological agents in the world. U1 is a deep breath, small nuclear ribonucleoprotein. And I know I said this was the best named thing in the universe. That right there is way too much of a mouthful. So folks in the business call little guys like this a snurp. He's a snurp. I don't care what you say. This fact alone is worth the price of admission. I love the words in this discipline. How could you not? And say that out loud. Snurp. Snurp. It is physically impossible to talk about snurps without a little smile on your face. They're so great, and they do such nifty work. But let's get down to business. The protein scaffolding in U1 is organized around this core piece of RNA. Check this out. This coil of RNA letters is directly complementary to the code at this end of our intron, the 5' splice site, which means U1 can bind to our pre-mRNA at the exact right space at the exact right time. This is the incredible power of RNA. We can use RNA workers like U1 to simultaneously bind to chemistry and information. But let's just see this in action first. Let's edit our precursor mRNA and get out of here. Step 1, we gotta assemble the spliceosome complex itself. It starts off simple. U1 actually binds to the 5' splice site during elongation and then goes on to help assemble the full spliceosome as elongation is playing out. Splicing mRNA actually happens at the same time RNA is being transcribed. And you know what? One day I'll have enough GPUs to render transcription and splicing happening at the exact same time, but if I tried that now, I would probably burn my house down. Speaking of which, if any of y'all know somebody at NVIDIA who could hook me up, I would really appreciate it. I got big ideas, and boy howdy am I ever ready to sell out. Either way, for now, let's just be cool with animating these processes separately. After U1 binds, the U2 snurp is going to get recruited to the other side of this intron. U2 will bind to the branch point of this intron. Then we're going to meet the next 3 snurps all at once. U4, U5, and U6 here are better known as the tri-snurp, because there's 3 of them. The tri-snurp and U2 are going to do the actual splicing business once the tri-snurp gets into position right about here. So the first big thing that happens is U1 will hand off the 5' splice site to the tri-snurp. U4, U5, and U6 will work with U2 to do the actual business of splicing. U1's job is done now and will mosey off to find another intron further down the transcript. We're now ready to get down to the real business of splicing. This full spliceosome will use ATP to reshuffle some pieces to fully incorporate the 5' side of this intron. That'll eject some pieces of the U6 snurp and pretty much all of the U4 snurp, which will help wrap this intron around the catalytic business end of the spliceosome. All this is going to set the stage for our big player, the NTC protein complex, which will bind to basically where U4 left and form the floor of our almost fully activated spliceosome. The active site is now fully ready to go. We just need to push the 2 ends of this intron together so they'll bind to each other instead of our exons. To do that, the spliceosome will reshuffle itself even more to get these 2 ends of the intron closer together. And finally, the spliceosome will snap into a much tighter conformation, finally putting our 5' splice site close enough to this branch point at the other end of the exon. This is the perfect configuration for the first real step of the splicing process, which is called branching. Basically, our 5' splice site will stick itself to the branch point on the other side of our intron, which means that down here, 1/2 of our exon has been completely freed from the intron, but up top we still have another intron bond to cut. So we're going to shove these guys even closer together, and this is going to be tight enough that both halves of our exon are going to want to be together way more than they want to be with that intron bond. So they'll snap together while this looping intron, called the intron lariat, just kind of gets snapped off. Our exon is ready to go while our intron is ready for recycling. And changing the view real quick too, let me pull this part of the spliceosome open to show you our freshly spliced mRNA transcript. Nice. We're going to send that off for final processing, while the rest of the splicing machinery disassembles itself ahead of the next reaction. And I mean, it was cool, I guess, to see that in its full molecular detail, but for my money, that was a little bit hard to follow. One day I'm going to make a massive video explaining all of these different colored subunits and all of the very cool and very deep organic chemistry that's happening here. But let's dial the complexity down a little just to illustrate an introduction to what's going on. And we'll just show the business end of the splicing reaction. No U1 handoff, just U2 and the trisnurp, cutting an intron out. So let's take it from the top. Again, U1 is going to bind to our 5' splice site, while U2 grabs the other side of our intron. U1 is going to bring its side of our intron to the trisnurp and then, bail. The trisnurp and U2 are then going to churn through a whole bunch of ATP so they can rearrange themselves enough to push the two ends of this intron together, bringing us to the actual splicing reaction, which has, again, 2 steps. Step 1 is called branching, where our 5' end gets shoved onto the branch point of the other side of this intron. Step 2 is exon ligation, where we'll squeeze this whole thing together even harder so our 2 exon pieces get glued together. The exon will then leave, and the intron lariat will be left behind to get chopped up and ready for another round of transcription. Nice. But I want you to understand, of course, that we skipped over a lot of really amazing details and really amazing chemistry, and to fully explore the true beauty of the human spliceosome, we're going to need like 2 more videos. Those are going to come way later, but it doesn't matter because we finally did it. We really went from DNA to mRNA in a eukaryotic cell. Awesome. But now that we've done it, that's the what, that's the how, that's cool. Go off and do your protein business, mRNA. Why did I put myself through this? Why did you subject yourself to nearly an hour of this jargon? We haven't covered the why of the spliceosome. And I mean, really look back at the whole splicing process. That is a ton of ATP we just used to cut out a useless sequence that, maybe just doesn't need to be there in the first place? And it gets even more expensive once you realize that most genes don't have 1 intron. We did this extraordinarily simplified version of splicing just so my computer wouldn't explode rendering this. In reality, the average gene has about 11 introns to edit out, and extreme ones can require dozens of splicing events. Why on earth would you sink this much effort into something this convoluted? Oh my god. Did evolution saddle us with runaway bureaucracies? Are we secretly inferior to our more streamlined and simplified prokaryotic cousins? This is a big moment in your personal educational journey as you come to respect the complexity in the deeper life sciences, because this gets back to how most of us usually learn the central dogma and how that can create the wrong impression of how life really operates. You learn that DNA gets turned into mRNA, which eventually will get transcribed into protein. Back in the day, we kind of treated RNA as this annoying kid brother to DNA, just a silly intermediary between the architect and the actual worker. But I can tear that whole idea down by letting you sit with a little math for a second. In order to accomplish the business of being you, your ribosomes synthesize anywhere from 75,000 to maybe as many as 500,000 distinct protein workers. That's called your proteome. It takes that much molecular diversity to accomplish pretty much anything. DNA is going to code for those proteins, right? So, riddle me this, hotshot. How many protein-coding genes are you working with in your DNA right now? Lock in your answers in the comments or something. And don't worry, I'm not going to drag this out. I'll tell you the real answer. It's around 20,000. 20K. And if you're like me, that juxtaposition is going to start blowing your mind. 20,000 genes yields upwards of 100,000 different proteins, maybe 500,000. But how? It starts getting a little existential almost. Wait a minute, my DNA only codes for, at best, less than 1/3 of the proteins I'm working with? So where does the rest of the information come from? If the central dogma is about that information, I'm missing a bunch of that information. Where is it? And the answer here is so great. Here's the thing. The spliceosome isn't a one-trick pony. It's not gonna splice just every intron it sees. Nah, friend. Depending on what cell type the spliceosome is in, it'll end up cutting big chunks out of its pre-mRNA transcripts, sometimes taking multiple exons with it, which means one gene can yield dozens of different proteins. The classic example used to illustrate this is the alpha-tropomycin gene, which codes for a super useful coiled protein that helps muscles contract. The gene coding for this protein has a good dozen or so introns, and depending on how you cut those out, you'll end up with 5 completely different proteins. One great for helping skeletal muscles contract, one better suited for smooth muscle, a handful that help fibroblasts, and one that's actually a critical piece of brain architecture. One gene, 5 different proteins with very different functions, all because the spliceosome can pick and choose exactly where to splice. This is an amazing trick because it means you don't need to have 3 to 4 times the amount of DNA you have in order to be you. And if you go back to what I just said, all 5 of those proteins are inside different cell types, which means you've got fine-tuned spliceosomes for whatever cell type you're in, which is one of the foundational mechanisms that allows you to be multicellular. Which means this is the fountain of what makes life so rich as a eukaryote. Because I'm sure you're wondering, how does the spliceosome know it's in a skeletal muscle cell and it has to cut up alpha-tropomycin the skeletal muscle way? Different cells express different non-coding RNAs, which will work with a bunch of other factors to control what splice site the spliceosome is looking for. And that's why we study the complex stuff here, because maybe an hour of jargon later and we have blown the doors off of some of the more oversimple popular assumptions about life. DNA is a very important part of what makes you, you. But you are not a robot managed by your DNA. DNA is not destiny, and instead, your whole enterprise is an incredibly complex DNA management problem. Different cells loosen up their chromatin in different ways, only allowing RNA polymerase II to accent certain cell-specific genes. And different cells express different regulatory proteins and small nuclear RNAs that guide and manage the spliceosome, making it so that even though your white blood cells and stomach lining all have the exact same DNA, they can still take on the structure and functions that they need without skipping a beat. RNA isn't just some lame intermediary, RNA might just be the whole ballgame. And we're only right now just starting a journey that will allow you to see how much complexity this little guy can create. But we've managed to go on one complete journey here, and for now that is enough. Here's what I want you to take away from this video. Here's why I sank months of my life into animating what is ostensibly a laundry list of molecular assistants. The majesty in life is not in the thing itself, but in all the infinitesimal ways it is coaxed and coordinated. RNA polymerase II and other polymerases are incredibly powerful, yes, but this army of transcription factors pushes and prods and gradually guides this unstoppable force of molecular will into something beautiful. Throughout this series, we're going to meet a near uncountable cast of managers and side characters. And gradually, if you stick with me, you'll start to see the stumbling, incremental hand of evolution at play behind the scenes, and the remarkable plasticity that makes life so powerful. It is that adaptability and plasticity that has helped us take over this planet and ascend as high as we have. You'll see a molecular conspiracy that fought and scrambled its way to complexity, and that's where the awe truly lives. It is frustrating, all the effort it takes to learn and how much effort it takes to unravel the molecular secrets that guide our cells. But then all the effort starts to cohere, and you start to see how much this good earth conspired just so your consciousness could emerge out of the darkness. Pretty much every iota of energy your body squeezes out of your food was once sunlight boiling out of the core of a star. But thanks to the gentle, subtle management of your metabolism, you breathe instead of burn. The complexity is what makes it worth it. We're only just beginning to unravel this thread. But for now, we've accomplished what we set out to do. We've gone from DNA to fully processed mRNA. This shrimpy little mRNA strand that we worked so hard to make is now bundled up and ready for action. It's time for this little fella to find one of those nuclear pores and head out to the next big phase of the central dogma. And hey, good luck, bud. Gosh, I'm so excited to see what you'll accomplish once you're out there in the big cytoplasm world. But let's let you go for now. We'll catch you in the next video, mRNA. It's been a great journey, but for now, let me just say, I really appreciate your time. Thank you so much for being here. And hey, thank you so much for reaching the end of the video. I know this one was a doozy, so I really appreciate you being here. If you made it this far, let me know what your favorite DNA letter is. I'm honestly curious. Mine's adenine, but whatever. If you like this video, please consider subscribing to this channel and joining us over at my Patreon. This year is going to be all about kind of revamping what we're doing on my Patreon as well, and making our tiers actually mean something, so make sure you check that out as I go through there. Another really important way you can support the channel is subscribing to our newsletter. Head over to watchclockwork.com, and you'll be able to get access to basically a monthly mini episode of Clockwork itself. I'm going to be revamping how we do videos for the next couple of months to help us get ahead a little bit, so you'll be seeing more of those shorter newsletter-style videos, as well as quicker topics. Part 3 of the Central Dogma is coming, but it's going to take a minute to get there. Either way, we have a lot of great stuff to explore this year. Thank you so much for being here with us. I really appreciate your time. And as always, I like to leave you with peace, love, and snurps. Everyone be well. Thank you so much.